Greetings. My name is Bhandari. I'm a PhD student in Dr. Babak Baban's lab in Department of Oral Biology and Diagnostic Sciences. The topic of my presentation is innate lymphoid cells as an immune maestro in tongue tumor. Starting with the introduction, tongue squamous cell carcinoma is the most common type of oral and maxillofacial malignant tumor. Currently, there are limited options for treatment for tongue squamous cell carcinoma, because of which the overall survival rate is only about 50%. This cancer contributes about 3% of the oral cancer in the United States. As of data of 2020, there were 377,700 cases of tongue cancer worldwide. This cancer is more seen among the males when compared to females. The risk factors include alcohol, smoking, infection with the human papillomavirus. Talking about the treatment modalities, like I said before, there are limited treatment modalities which includes primarily surgical and also radiation and chemotherapy. Moving on to brief introduction of cannabinoids. It's a compound that has been derived from the plant called cannabis sativa. This plant has more than 113 compounds, and among them the most active ingredients is tetrahydrocannabinol and cannabidiol, which is CBD. CBD has been used in ancient times for medicinal purpose. It has non-psychoactive property, unlike THC, which has psychoactive property. It has excellent medicinal properties. It has immune regulatory properties, because of which it could be a key target for therapeutic modalities for malignancies, including tongue tumor. And there is also increase in public and scientific demands for this compound. Moving on to innate lymphoid cells. So these cells, they are the growing family of the immune cells that mirrors the functions and phenotype of T cells. But unlike T cells, these cells do not have antigenic receptors, and rather they produce an array of proteins known as cytokines to produce an immune response in response to an original insult. There are three types of ILCs, which include ILC1, the transcription factor is TBET, ILC2, the transcription factor is GATA3, and ILC3, the transcription factor is ROR gamma T. These ILCs, they present high heterogeneity and plasticity, which make them able to either inhibit or promote the tumor growth. This differential response of ILCs to the tumor is a consequence of crosstalk between the cells and the different components of the tumor microenvironment. Moving on to the objective of the study, the objective is to dissect the crosstalk between the innate lymphoid cells, ILCs, and inhaled CBD in tongue squamous cell carcinoma. Starting with methods and methodology. This is a chemical carcinogen model, meaning a compound called 4-nitroquinolone 1-oxide was used to establish the cancer cells. This is a well-established model for cancer. So the mice that were used for the study was C57BL6. The age of the mice were 9 to 12 weeks old. These mice were randomly classified into two groups, 5 of each group, inhaled CBD and inhaled placebo. The cancer cells were injected into the right tip of the tongue of the mice at a concentration of 1 into 10 to the power 6. And the tumor was allowed to grow for the span of 8 days. Once the growth was seen on the tongue of the mice, the treatment was started. The inhaled CBD was given at a concentration of 10.8 mg per kg, that is 6 actuation per time point, for the next 8 days. And on the 9th day, the mice were euthanized and the tongue tissue and blood were collected for further analysis of flow cytometry and histology. In case of histology, hematoxylin and eosin staining was done to recognize the cellular pattern and tissue structure. And to identify or understand the characteristics, physical and chemical characteristics of the cell types, immune cell types, flow cytometry was done. In addition, the size and weight of the tumor was also measured manually after the euthanization of the mice. This is a picture explaining how inhaled CBD or placebo was given to the mice during the study. Moving on to the results, starting with the CT scan of the tongue. On the left hand side is a placebo, on right hand side is the CBD. There is a picture of a tongue close to this bar, as you can see. On the left hand side you can see dark circles or dark surface area. This is an aerial view, so you can see as like a raised portions in case of placebo, whereas in case of CBD you can see a smooth surface, homogeneous surface, and no capsules were seen. suggesting that not only the model was established, but also the use of inhaled CBD, it suppressed the tumor growth. We also measure the tumor weight in both placebo and CBD group, and what we found was with the use of inhaled CBD, the tumor weight was significantly less in CBD group when compared to the placebo group. And on the right side is the quantification of the tumor weight measured. Moving on to hematoxylin and eosin staining. This was done to recognize the cellular pattern and to identify the tissue structure. So there are four boxes on the left hand side. The upper box is the 1x and the lower box is the zoomed in pictures. So on the left hand side is the placebo group and on the right hand side is the CBD group. On the placebo group, you can see the darkness of tumor infiltrating the surface of the muscular fibers of the tongue or the skeletal muscles of the tongue. The cells were loosely oriented, poorly differentiated, and they lost their identity, and the tumor was spreading. Whereas with the use of inhaled CBD, the spread was halted by the necrotic tissue within the tumor itself. There were less cells in the extracellular matrix, and there were keratin pores seen more than in the placebo group, suggesting of that inhaled CBD use halted the spread and metastasis of the tumor. On the right hand side, you can see the surface area measurement and the quantification of the tumor, which we found that the surface area of the tumor was significantly lower in CBD group when compared to the placebo group. Now, flow cytometry was done to identify the characteristics of the immune cells. So ILCs, these are lineage negative. So we use lineage markers such as macrophages lineage, T cell lineage, and B cell lineage, and CD45, which is a universal pan marker for white blood cells. We use T-bet, GATA3, and ROR gamma T as a marker to identify ILC 1, 2, and 3 population respectively. Isotype was used as a negative control. So as we said, ILCs is lineage negative, so we only took the lineage negative and CD45 positive populations to see the ILC population. Well, we found that inhaled CBD use increased the ILC2 highest, followed by ILC1 and ILC3 respectively. So we come to a conclusion that CBD treatment not only promoted the activation and polarization of ILC, but they also seem to be effective contributors to the modulation of immune mechanism in tumor microenvironment. And this could be a potential immunotherapeutic target in tongue squamous cell carcinoma. So this warrants further research and to understand the ILCs and their property in tumor microenvironment in case of tongue tumor with the use of inhaled CBD. Thank you.